All right. Renal corpuscle.
Renal corpuscle. And we can see that a variety of tubules around here. Most look like they are proximal convoluted tubules, because they're kind of ragged, not very well fixed. That's kind of a clean border. That's clean, so that's probably distal convoluted tubule. Don't sweat that. Just realize why it's a smooth luminal profile. Don't have a lot of microvilli. When they look ragged and they're poorly fixed, it probably suggests they're microvilli. All right, so this portion of the renal corpuscle, glomerulus, and the surrounding boneless capsule. Right. All right. So if we think then about the glomerulus, it's the site of the filtration barrier, all right? And this is a pretty nice electron micrograph transition. We can see an erythrocyte here, so this is a capillary lumen. We can see little bits of endothelial cell cytoplasm, and notice we've got these pores or fenestra, or we say that the endothelium is fenestrated. A basal lamina. I mean, that's off the wall, to say basic lamina. Basal lamina produced by both these cells and the podocytes. And this is kind of the sieve, or filter, if you will. One interesting thing is that it does get clogged up. You know, you take a backpacking filter, you know, after a while it's no good. You know, it's all clogged up. Mesangial cells come in here, phagocytize this membrane, clean it up, if you will, and then it's laid down again. So there's a perpetual process of renewal. All right, we've got a renal corpuscle with histologic evidence of the justoglomerular apparatus. Do we see it? So clearly, let's start with what we know. Glomerulus. That kind of has a nice look of an afferent arteriole, right? But this puppy is the ringer, the macula densa, all right? And if you take the time, even at 4x, looking at your kidney slides, you know, scanning a bunch of renal corpuscles, you might come up with something like this, all right?
How do you know that's the macula densa versus just a regular?
The nuclear density, you know, that macular, you know, spot, dense spot. And the nuclei of the cuboidal cells are crammed right next to one another. Okay, here it looks like we have an afferent arteriole coming in. We've got a vascular pole, and I don't see very well, I don't see what I would call the macula densa there. Okay. We're in the medulla, right? No corpuscles. Remember that the fence, if you will, the arcuate vessels, arteries and veins, define that interface between cortex and medulla. So, what are these blood vessels? Exactly, vasorectal. And, you know, they start out as arterioles, and then they can branch into a capillary network, which then drains into a venular network that runs opposite, adjacent, to maintain that border. What about something like that? Thin loop of Henle. And you can see that this clearly is a squamous. That's probably the same thing. Here we've cut through the wall. It's kind of a tangential section. What about something like that? Or more particularly like that? Yeah, because you can begin to pick out the borders between adjacent cells. Now you're not going to see that in the thick descending or ascending sections because the basolateral infolding that's chock full of mitochondria obscures that. All right? This is a really nice micrograph that shows how abrupt that transition is. So they're saying, well, we know this is proximal tubule because we can see the microvilli. So this would be the thick descending or straight portion of the proximal tubule, right? And then it's a tangential section, so it's cut like that. And you pick up, look at the thin portion is certainly squamous right there. And same song, second verse for the ascending portion. There's a squamo-cuboidal junction would be the way to look at it. junction's funky, but an abrupt transition. Now you can see, look at all the mitochondria there. They're calling this a distal tubule, which makes sense. Lots of mitochondria, but look at the luminal profile. You don't have many microvilli. Try to get out of the What kind of epithelium? And realize that transitional epithelium from minor calyx all the way to the proximal portion of the urethra, including the ureter and the bladder. How are you going to tell the difference between ureter and bladder? Lots more muscle in the bladder wall than in the ureter wall. Same kind of epithelium. It's just one of those kind of obvious sorts of things. This image is, some of you may have this slide in your slide sets, others of you don't, and you don't see the epididymis. With that in mind, let me recommend again that you use that histology image bank. Some of you may not like it because I didn't put arrows and whatnot on everything, but I did that on purpose because if you click on the image, you've got descriptive text which describes features that should be there, and the intent was to help you master the vocabulary, all right? But this, for those of you that didn't see epididymis, whose slides were just testes only, you might want to go to the image bank. But you can see clearly the seminiferous tubules, these connective tissue partitions, the testicular parenchyma into lobules. You can see the tunica albuginea. Whether or not you have the vaginalis, which would be a mesothelial lining adherent, depends on processing. Probably not see it. Come back here into the mediastinum, and you can pick up the efferent ductule. So this, all right, let's back up. If we start with a seminiferous tubule, 1 to 4 per lobule, wound every which way. But imagine it as just a U-shaped tube, right? And the distal portions that are open are straight. They drain into a labyrinthine channel, the reedy testes, which then drains into 20 or so efferent ductules, which then coalesce into one tube for the epididymis. So the epididymis here is one tube highly coiled, all right? If we look at the seminiferous tubule, you get profiles like that. This looks like a reasonable cross-section, kind of longitudinal, kind of tangential. You've got a lumen, you've got this PAS-stained basement membrane, which in animals, as Nigel said, can have a little bit of smooth muscle, not so in humans. And then you've got the seminiferous epithelium. Two types of cells we need to be concerned with. These spermatogenic cells, spermatogonia, and primary and secondary spermatocytes. I don't care that you can identify what stage. What I do care is that you realize that there are meiotic reduction divisions going on there, because the gametes are haploid, right? Half the amount of chromosomes. The spermatogonia are easy cells to pick out. They're the round cells that are sitting right on the membrane. More difficult to see are the Sertoli cells, which are very, very important because they provide that blood-testis barrier and all sorts of nutritive functions, as well as crank out androgen binding protein, which keeps the testosterone produced by interstitial cells of Leydig in the vicinity so you can have spermatogenesis going on. All right? The way you pick those cells up are basically by finding their nuclei. Because these are metabolically active cells that are cranking out things, the chromatin is euchromatic, which means it's light staining, except for the nucleolus. All right? That's in contrast to the spermatocytes. All they're worrying about is condensing, forming chromatids, condensing, dividing, that sort of thing. Nice example of a Sertoli cell. Here's one also. We don't see the nucleus. There's one. There's one. And actually, I should say Sertoli cell nuclei, right? Bam, bam. All right? Right there. Another one right here. Interstitial cells.